Hello and welcome. Today let's talk Chapter 3. Chapter 3 takes us into the cell structure and function. And we'll start with 3.1. This takes us into the cellular level of organization. So when we talked about the hierarchy, so here we're going to now the cellular level. Now I'd like you to know that the cell marks the boundary between the non-living and the living. The molecules that serve as food for a cell and the macromolecules that make up a cell are not alive, yet the cell itself is alive. When we talk about the cell, I'd like you to know that the cell is the structural and the functional unit of an organism. It is the smallest structure capable of performing all the functions necessary for life. There's three different types of cells that exist in nature, and you will be familiar with two of the more common ones, and those three include prokaryotic cells, eukaryotic cells, and archean cells. The two that you are going to be more commonly familiar with will include the prokaryotic and the eukaryotic cells. The prokaryotic cell is commonly called bacteria, and it lacks membrane enclosed structures, such as the nucleus we'll see. And the second type are eukaryotic cells. When we talk eukaryotic cells actually possess membrane enclosed structures. Not only will we go through those structures, but we're also going to see what they are all doing for the cell and eventually for the whole body. Next, when we talk archean cells, a little bit I'll tell you there, they possess qualities of both prokaryotes and eukaryotes. So you can think of cells that have, again, qualities of both prokaryotes and eukaryotes, kind of like a mix. Now, when we go through and we talk about the basic components of eukaryotic cells, the basic components of eukaryotic cells is what we'll discuss in this chapter. And in order to view these components, a microscope is required. A microscope is going to be required. Next, let's talk about the cell theory. The cell theory states that all organisms are composed of one or more cells. The cell theory states also that cells are the basic living unit of structure and function in organisms. And third, it states that all cells come only from other cells. All cells come only from other cells. So that is our cell theory. Next, let's talk cell size. When we talk the size of cells, cells are quite small and they can vary. They can range from 1 millimeter. They can range in size from 1 millimeter down to 1 micrometer. Down to 1 micrometer. We'll look at an image in a few seconds, but a frog's egg is about 1 millimeter in diameter. It's large enough to be seen with the naked eye. If you've ever dissected a frog and you happen to have a female frog that was pregnant, you're able to see all the eggs right inside the abdominal cavity. Okay, most cells are smaller than 1 millimeter. Some cells, again, are as small as 1 micrometer. 1 micrometer. So here, this figure, it outlines the visual range of the eye. You can see the eye is able to see down to a little less than 1 millimeter, which we described as being the size of a frog egg, okay, down to a human egg. Next, then, it describes, you can see here, it outlines, again, the visual range of light microscopes. And then third, electron microscopes. And then here you can see the size of each of the structures. And they're giving you a nice picture there as well to get a nice visual. So when we talk cell size, cells being small is an advantage. It's an advantage for multicellular organisms. Why, we'll see? Because nutrients, such as glucose, can enter the cell. Waste, such as CO2, is able to exit out of the cell. Therefore, surface area affects the ability to get materials in and out of the cell. Now, as cells increase in volume, you'll see the proportionate amount of surface area is going to decrease, which is not then good for the cells. All right, let's talk 3.2 prokaryotic cells. Now, when we talk prokaryotic cells are going to be cells that lack a nucleus. Okay, cells that lack a nucleus, because we compared them to eukaryotic cells, which we said have a nucleus, and we'll look at that even further as we progress. Now, the domains archaea and bacteria are going to consist of prokaryotic cells. So it comprises domains, bacteria, and archaea. Prokaryotic cells, okay, generally unicellular organisms. They may be single, they may be found as strings, or these prokaryotic cells can be found as clusters. And not all bacteria are going to be disease-causing. Some are very beneficial commercially to the environment, to our bodies. In the intestines, we have gut bacteria, right, that helps with digestion. We've got bacteria in other places in the body as well. It's all beneficial bacteria there. Next, then, when we move through, I'd like you guys to know prokaryotic cells have a plasma membrane. They have a plasma membrane. Basically, all cells are going to be surrounded by a plasma membrane, we'll see. Since we're talking prokaryotic cells, we'll see they have a plasma membrane which surrounds the cytoplasm. Now, this plasma membrane, it forms a boundary that's going to separate the contents of the cell from the surrounding environment. Also, it's going to regulate the passage of molecules into and out of the cytoplasm. Now, cytoplasm, we keep saying, cytoplasm, I want you to know, okay, is the semi-fluid medium inside the cell. So the semi-fluid medium inside of a cell, we call that cytoplasm. And that cytoplasm itself, we'll see, is made up of water, salts, and dissolved organic molecules. Now, when we go through and we talk about bacterial anatomy, you'll see they have a cell wall. So we'll go through and we'll check out. Here we could see that phospholipid bilayer, the membrane, the plasma membrane we discussed. Here you can see it's made up of lipids. And here you've got the proteins in orange. And these proteins are what's going to regulate the passage of molecules into and out of the cytoplasm. So again, when we talk about bacterial anatomy, you'll see they have a cell wall. The cell wall is going to protect them. It's located outside of the plasma membrane, consisting of peptidoglycan. Some bacteria can even have a capsule around them. So now actual bacteria and its structure will all vary, and here we'll just go through and we'll mention a few components we can find in relation to the bacteria. So a capsule is going to further protect them, and this capsule is a gelatinous sheath that surrounds the cell wall of some bacteria. Flagella can also be found in some bacteria. Flagella, we'll see, is going to be basically helping the bacteria move. It's a thin, long appendage. It's used for movement in some bacteria. Fimbriae we can find in some bacteria. Fimbriae we'll see are going to be short appendages that help the bacteria to attach to appropriate surfaces. So it helps bacteria to attach to surfaces. And we'll look at all of these structures in a few minutes. we'll see they also have nucleoid, single chromosome basically inside the cytoplasm. It's a region of the cytoplasm where a single bacteria chromosome is located. And this region is not surrounded by a membrane. It is not itself surrounded by a membrane. We're also going to be able to locate ribosomes. Ribosomes, we'll see, they're going to be used for protein synthesis. They'll be used for protein synthesis. Thylakoids can also be found, which are going to be membranes of flattened discs, and they're going to contain light-sensitive pigments, and we're going to find that in cyanobacteria. We'll find that in cyanobacteria. So here you can actually see, first, the plasma membrane, and then surrounding that we said is a cell wall. And then some can contain this capsule. And then these fimbriae you see all around. And then this one flagellum. And you can see one flagellum. And then here under the microscope. And you'll appreciate more E. coli pictures when you get to microbiology. So here's a little table comparing archaea cells to bacteria and eukaryotes. So here you can kind of understand what the archaea will have, and then bacteria we just went through. And then let's go through eukaryotic cells. Let's talk about eukaryotic cells. Eukaryotic cells, they're quite complex structures. Their Their main distinguishing factor is their nucleus. Their nucleus is going to be found separating the genetic material from the cytoplasm. It separates the genetic material from the cytoplasm. Eukaryotic cells possess a variety of other organelles also, besides the nucleus. Cell walls can be found, we'll see in some eukaryotic cells. So some eukaryotic cells have a permeable but protective cell wall in addition to the plasma membrane. But most of the time it's absent. Plant cells, for example, they will not only have a primary cell wall, but they also have a secondary cell wall. So plant cells have a primary and secondary cell wall. And eukaryotic cells, so they possess organelles, membrane-bound organelles. And eukaryotic cells not only make up animals, plants, fungi, and protists, if you recall. And protists. One way I remembered eukaryotic, you. I was in the mirror talking to myself. I say, you have the eukaryotic cell. Obviously, I'm not bacteria, nor am I archaea. Okay, but at this stage, it usually just all seems overwhelming. Okay, so one way to kind of throw that one out of that worry box of your mind. Okay, eukaryotic cells make up animals. That's you and I. We're all animals. Plants, fungi, okay, and protists. Let's move and let's talk then about cell walls. So cell walls, I told you, some eukaryotic cells have cell walls. Plant cells have a primary and a secondary cell wall. Cellulose they have, and chitin are going to be found making up fungi cell walls. Cellulose and chitin are found making up fungi cell walls. And we'll talk more about that towards the end of the semester. Also found in algae, protists, cell walls. Cellulose and chitin. Lignin is also found in secondary cell walls. Now, when we talk fungi cell walls, some cellulose and some chitin. And some chitin is in there. Now, we talk organelles of eukaryotic cells. When we talk about the organelles of eukaryotic cells, okay, can be thought of as a factory. I want you to think of, when we think of eukaryotic cells, as a factory. It's a factory that produces things, right? Factories produce things. So this factory, these cells inside of us, okay, what are they producing? They're going to be responsible for producing, we'll see, lots of proteins. That's going to be the main thing we're going to focus on, proteins. Now, these organelles of a cell operate at the same time, just as assembly lines do in factories. So you think of them as little miniature factories, these cells. Now here we could go through and we can talk about the main structures that are going to be making up the eukaryotic cell. And this is what we're going to go through in this chapter and look at in greater detail. So first, here we can appreciate all of those structures now, not by list, but actually visualizing them as a picture of a cell. So here we can see a few structures: the cell wall, the plasma membrane, the nucleus, nucleoli, ribosomes, endoplasmic reticulum, or we can abbreviate them as the ER, rough ER and a smooth ER, which are the two subdivisions of the endoplasmic reticulum, because there's two types of ERs, a rough and a smooth ER. Golgi apparatus, we'll check out lysosomes, vacuoles, and vesicles, peroxisomes, mitochondria, chloroplasts. In relation to plant cells, we'll look at, because then we're going to get into photosynthesis, the cytoskeleton, cilia and flagella, and then the centrioles. So here you can actually appreciate those various structures. Plasma membrane, right on the outside, nucleus, nucleoli, rough endoplasmic reticulum, okay? You can see here very nicely, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, peroxisomes, okay? Polyribosomes you can see in here, other structures, okay? Vesicles we've gotten here. And then the cytoplasm is all this area right inside of here that contains all of these structures. So basically everything inside of this plasma membrane, this is all cytoplasm, and the cytoplasm contains all these various organelles within it. When we zoom in on that plasma membrane, again, we saw what it looked like, and you saw the proteins in it before also. Here's a plant cell. Here you could see the cell walls, okay? And then we've got inside of here the plasma membrane, cytoplasm inside. And then here you can see the nucleus, nucleolus, ER, rough and smooth, Golgi apparatus, chloroplasts. We don't have those in our cells. Okay, mitochondria you can see, and you can see the central vacuole. Again, we don't have, but they do. Again, eukaryotic cells, both eukaryotic cells. But animal cell, plant cell. Animal cell, plant cells. So compare and contrast the two. First organelle we want to talk about and we want to discuss is going to be the nucleus. The nucleus. The nucleus is a prominent structure with a diameter of about 5 micrometers. With a diameter of about 5 micrometers. In photographs or in slides, the nucleus looks like a dark softball. It looks like a dark softball that's found within the cell. It looks like a dark softball found within the cell. It's an important component of the cell. And the reason why it's an important component of the cell is because it stores genetic material, I told you. It stores DNA. Stores DNA. In relation to the factory analogy I gave you guys, the nucleus you can think of as the head office. You can think of it as the office, the head office. DNA is going to decide, you'll see the characteristics of a cell. It decides the characteristics of a cell and its metabolic functioning. Now, when you look in the nucleus, you don't just see a DNA molecule sitting there. What you see is chromatin. Chromatin, it consists of DNA and proteins. It's always found scattered throughout the nucleus and undergoes coiling to become highly condensed structures called chromosomes. It undergoes coiling and condensing to become chromosomes. And we're going to see when that happens. That's going to happen when the cell is ready to divide. If the cell is not dividing, how is that genetic information going to be found? As chromatin. As chromatin. Chromosomes are basically a single piece of coiled DNA. And I want you to know human cells contain 46 chromosomes, or 23 pairs, we can also say. 46 total chromosomes, or 23 pairs. Also found within the nucleus, is a nucleoli. So also found inside the nucleus are nucleoli, where ribosomal RNA is made. And let's look at that. You could think of that as a little golf ball within the softball, the nucleolus, right inside of there. Now, the nucleus is separated from the cytoplasm by a double membrane that's called the nuclear envelope. So the nuclear envelope is a double membrane that's going to separate the nucleus from the cytoplasm. This nuclear envelope is continuous with the endoplasmic reticulum. It is continuous with the endoplasmic reticulum. The nuclear envelope has nuclear pores. It has nuclear pores on it. These nuclear pores are going to permit bidirectional transport. They will permit bidirectional transport of proteins and ribosomal subunits. Here you can see that. So what we've done is we've highlighted the nucleus, and here when we highlight the nucleus, you can actually appreciate all of that chromatin inside of here, besides the nucleolus. And then here you can appreciate that double-layered nuclear envelope, and then all of its nuclear pores that it has on it. And then inside here, we have nucleoplasm. Nucleoplasm. Like we had cytoplasm in the cell, here we have in the nucleus nucleoplasm. And nucleoplasm is a semi-fluid medium in the nucleus. A semi-fluid medium in the nucleus. I want you to know this nuclear envelope. This nuclear envelope is going to disappear during cell division. During cell division, this envelope disappears. This envelope disappears. Why does it disappear, you're going to ask? Because we need to let this genetic information basically out. So that's when these the chromatin you see here becomes chromosomes. And then those chromosomes are going to have doubled, and we need to let the one pair out so it can go to the next cell. But we'll look at all of that as we progress throughout the semester. Interesting stuff in here. Interesting stuff. And then here, let me show you how it is continuous. You can see right in here. The nuclear envelope is continuous, I told you, with the endoplasmic reticulum. In this picture, you can appreciate that. You can see how the outer layer makes its way out, and then it forms the endoplasmic reticulum. Let's talk ribosomes. When we talk ribosomes, ribosomes are responsible for the synthesis of proteins. They are responsible for the synthesis of proteins, and ribosomes will use messenger RNA. They will use mRNA as a template. They'll use messenger RNA as a template. We'll go through, we'll check out all of that stuff. I'm so excited. I can't wait till we get there. When we talk ribosomes, I want you to know ribosomes are composed of two subunits. They are composed of two subunits, and the two subunits are called a large subunit and a small subunit. You'll learn more about those when you get to micro. You're going to actually have to give them names, and the names are numbers, 50S, 30S, and whatever you have there. So they're composed of two subunits that are called a large subunit and a small subunit. And the reason why is because of their size. Ribosomes can be found individually, as we see right off to the side here. If you look in detail, some ribosomes here, they can be found individually in the cytoplasm or as groups. Or they can be found, as we see here in this picture also, attached to the endoplasmic reticulum. So they can be found individually in the cytoplasm or as groups, or they can be found then attached to the endoplasmic reticulum. So let's look at the endomembrane system in greater detail. Now, when we talk about the endomembrane system, it consists of the nuclear envelope that we went through and we talked about, the endoplasmic reticulum we're going to go through and we're going to look at in greater detail, and then also the Golgi apparatus, and included there will be also tiny vesicles. The little vesicles, we see little membranous sacs. First, let's check out the endoplasmic reticulum. Now, when we talk about the endoplasmic reticulum, I'd like you to know that the endoplasmic reticulum is a complicated system of channels. It's a complicated system of channels and sacs. It is physically continuous with the nuclear envelope as we have described. The rough ER, the rough endoplasmic reticulum, contains ribosomes. So hence the name rough. It's studded with ribosomes. The rough ER contains ribosomes which synthesize proteins and send them to the interior of the endoplasmic reticulum, where then they are processed and modified. Next, then we have the smooth ER, the smooth endoplasmic reticulum. The smooth ER is continuous with the rough ER. It is continuous with the rough ER. This area, smooth ER, has no attached ribosomes, hence the name smooth ER. So here you can see smooth ER and then rough ER. So this area has no ribosomes, hence the name smooth endoplasmic reticulum. The smooth ER synthesizes the phospholipids found in the cell's membrane. In the testes, so you'll see its functions are going to vary when you go from cell to cell. In the testes, we'll see smooth ER produces testosterone. In the liver, smooth ER is going to be responsible for detoxifying drugs. It detoxifies drugs. And in muscle cells, it's going to be responsible for storing calcium. It will be responsible for storing calcium. Smooth endoplasmic reticulum also forms vesicles. It forms vesicles. Smooth ER also forms vesicles in which products are transported to the Golgi apparatus. Now, when we talk the Golgi apparatus we'll see is going to consist, the Golgi apparatus is going to consist of a stack of sacs. It's going to consist of a stack of sacs. It looks like a stack of pancakes, I like to think of it as. Here you can see one stack, another stack, another stack, another stack, another stack, another stack. A stack of pancakes or a stack of plates. Either way. It's referred to as the shipping center. The Golgi apparatus is referred to as the shipping center. It's the shipping center of the cell. The reason why it's referred to as the shipping center of the cell is because it works like USPS, the United States Postal Service, or UPS, or FedEx. The reason why we say that is because it receives, it collects, it sorts, it packages, and it distributes materials. Just like those three companies do. The Golgi apparatus, it receives proteins and also lipid-filled vesicles. And you can see that here on both sides. So it receives proteins and also lipid-filled vesicles that come from that rough and smooth ER, that come from the endoplasmic reticulum. Proteins and lipids are modified here, and they are repackaged into secretory vesicles. And they are packaged into secretory vesicles which proceed to the plasma membrane to discharge their contents, or can even stay inside the cell if the contents are meant for the inside of the cell. So that's the Golgi apparatus. I want you to make sure you go through and understand each one of these organelles' functions. So here you could see the nucleus, rough ER, okay, and then some contents making their way right from the rough ER to the Golgi apparatus, while others make their way to the smooth ER, and then from the smooth ER to the Golgi apparatus. And the Golgi apparatus does its thing, and then prepares them for exit. Next, then let's talk about lysosomes. Lysosomes are vesicles. They are vesicles produced by the Golgi apparatus. Vesicles produced by the Golgi apparatus. Lysosomes, I'd like you to know, contain digestive enzymes. They contain digestive enzymes. You could think of the lysosomes as the garbage disposals of the factory. You could think of them as the garbage disposal of the factory. When we talk about lysosomes, they contain hydrolytic digestive enzymes. And what they do is they break down unwanted foreign substances or even worn out parts of cells. And they also can bring macromolecules into the cell. Next is vacuoles. When we talk about vacuoles, they're going to be found in plant cells. They're large membranous sacs, larger than vesicles. And what they do is they store substances. They store substances such as water, pigments, or toxins. Let's talk peroxisomes. Now, when we talk peroxisomes are similar to lysosomes. They are similar to lysosomes because they're going to contain enzymes. And peroxisomes, when we talk about, they're similar to lysosomes, and they're vesicles again, that contain enzymes. Now we talk these enzymes. Action of their enzymes are going to lead to the production of hydrogen peroxide. Hydrogen peroxide then is toxic to our cells, right? That's why we don't put hydrogen peroxide on our cuts and wounds. Okay, the actions of peroxisomes, enzymes, are going to lead to the production of hydrogen peroxide. Hydrogen peroxide then is toxic, so hydrogen peroxide quickly gets broken down into water and oxygen. Again, because of it being toxic to the cells, it quickly breaks down into water and oxygen thanks to the enzyme catalase. Thanks to the enzyme catalase. Now, when we talk about the functions, functions are going to vary amongst cells. In liver cells, peroxisomes are going to metabolize fats or produce bile. In germinating plant cells, they oxidize fatty acids. So here we can appreciate a peroxisome in greater detail, but let's move back. Do we have any here? Let's jump back to this picture right in here, where we can see an abundant amount of peroxisomes right here, right in there, right in here. Another one, I think, right in, yeah, another one right here. Okay. Next, let's move down to our energy-related organelles. Our energy-related organelles. Now, when we talk energy-related organelles, I want you guys to know many organisms carry out cellular respiration. Many organisms carry out cellular respiration, which is the process by which chemical energy of carbohydrates gets converted to ATP. Now, this process is going to take place in the mitochondria. It takes place in the mitochondria. So when we talk about mitochondria, I want you to know, are going to be the power plants, the powerhouse of the cell. Substrates are broken down in the cytoplasm, and then they get transported into the mitochondria, and then they're converted into ATP to be used by our cells. Mitochondria divide by splitting. And they do that to fulfill the body's need of ATP. They're really, really awesome organelles. Now, when we look at the structure of a mitochondria, you can see the structure of a mitochondria here in greater detail. So here you can see you have an outer membrane, and then you have an inner membrane. The inner membrane actually goes all the way to the center of the mitochondria, and then comes back down, and then goes back inside and comes back down, goes back inside towards the center, comes back out to the side. Right? That's all of its structure. Now, when it goes to the inside, these little folds it forms here are called cristae. And these cristae are invaginating into the matrix. The matrix is a space right here in the center. So here you can also see chloroplasts. And then the inside of the chloroplast I'd like you to know about as well, because we're going to talk about that when we get to photosynthesis in greater detail. Here you can see you've got an outer and inner membrane. And then here we can see we've got basically grana. Okay, these are, you can see thylakoids. And then a stack of thylakoids we refer to as a grana. We'll talk about that again as we progress. And inside you can see the thylakoid space. And we're going to look at that in relation to photosynthesis. And then in the center, you can see of this chloroplast, you've got stroma. So these are a couple of energy-related organelles. Now, when we go through, we talk mitochondria. Okay, so we've found in all eukaryotic cells. Okay, site of cellular respiration, we said. Bounded by double membrane, we saw there. It has a matrix, the cristae, invaginations. They help to increase the surface area of that inner membrane. And mitochondria, they contain their own DNA. Mitochondrial DNA is what it's called. Mitochondrial DNA. So beautiful picture. And here they're showing a good picture where you can see photosynthesis basically how here they're showing a nice picture of how these energy related organelles function. We'll talk photosynthesis and we'll talk cellular respiration. So we took care of chloroplasts, mitochondria. Let's talk cilia and flagella. Cilia and flagella. Now when we talk cilia and flagella, these are examples of microtubules. Cilia and flagella, they are hair-like projections that can move like a whip or an oar. They can move like a whip or an oar, and they will move like a whip or an oar to aid in cell movement or to help move things along the surface of the cell. Either to help in cell movement, moving of the cell, or to help move things along the surface of the cell. In eukaryotes, cilia are much shorter than flagella. However, I want you to know both are membrane-bound cylinders. Both are membrane-bound cylinders. They have a 9+2 pattern of microtubules arrangement. The arrangement of their microtubules is in a 9+2 pattern, and I'll show you that in a few seconds. So some cells that have flagella are capable of moving. Example, sperm cells. Now the cells that have cilia, example would be the cells that line the respiratory tract. They have cilia, and cilia is going to sweep debris back up into the throat. Cilia will sweep debris back up into the throat where it can be swallowed or ejected. And what this does, it helps to keep the lungs clean. Cilia are much shorter. Cilia are shorter than flagella. The cylinders are composed of a 9 microtubule doublet arrangement in circles around 2 central tubules. And this is exactly what we see there. So 9+2 arrangement, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9. Surrounding, we said, 2 central tubules. Surrounding 2 central tubules. So we look at this in greater detail, we see this image right in here. And then here you can appreciate in greater detail how those tubules look themselves. And again, they require ATP too.